professor of endocrinology at British Columbia University in Vancouver, and who shows that in foam cells in atherosclerosis, most of them are coming from smooth muscle cells. Welcome.
Merci Mathilde pour la gentille introduction et aux organisateurs pour l'opportunité de partager nos résultats avec vous. Je voudrais continuer en français, mais mon français n'est pas assez complet, alors je changerai maintenant à anglais. J'espère que vous me comprendrez. Et pouvez-vous m'entendre à l'arrière? OK, merci. Alors, I have no disclosures. So the image of atherosclerosis that most of us are used to seeing is shown here, where it's proposed to be primarily a response to injury, with monocyte-derived macrophages being the primary drivers of atherosclerosis. And there is really not much role for smooth muscle cells. And even in advanced lesions, the only role indicated for smooth muscle cells is forming the fibrous cap. And I think the reason we have come to accept this model is because most people have used mouse models of atherosclerosis for the last 30 years. And mouse atherosclerosis is most definitely a response to injury model. And mice do not have smooth muscle cells in their intima at the outset. And as Dr. Simeoni showed you, in the human intima, we have development of a thick layer of smooth muscle cells, which starts even in utero. And Virchow, as early as 1858, in the early days of microscopy, clearly illustrated that in aortic atherosclerosis, most of the foam cells appeared to be in smooth muscle cells in the deep intima. And this was also reported by other early investigators. Rudolf Altschul reported in 1950 that foam cells appear to arise primarily from smooth muscle cells. And Gear, McGill, and Strong in 1961, using early electron micrographs of human lesions, showed that in grossly normal intima, almost all the cells in the intima are smooth muscle cells, with very few histiocytes, which include macrophages and dendritic cells. And in early fatty streaks, the lipids were almost exclusively in smooth muscle cells. And Katsuda et al., including Russell Ross and Alan Gown, published in 1992, this is an 18-year-old male who had died from non-cardiac causes, that the intima shown here had macrophages primarily in just under the endothelium, and in a bigger magnification in the deeper intima were these cells with granules in them that are thought to be lipids and that stained primarily with smooth muscle actin. But this was around the time that people stopped looking at human lesions and primarily focused on mouse lesions. Nakashima et al. In 2007 published a very elegant study showing that here again we see these are all individuals' right coronary arteries, and the ages are shown. Some have died from cardiac and some from non-cardiac causes. And here you have the diffuse intimal thickening, but no lipid deposition and really very few macrophages. And then in a grade 1 fatty streak, you start to see some lipid deposition, mostly in the deep intima. In a grade 2 fatty streak, there's increased lipid in the deep intima. And in another grade 2 streak, again, lipids mostly in the deep intima, but progressing into the more proximal intima. And the reason that the lipids are thought to deposit there is that LDL and other atherogenic lipoproteins can diffuse through the endothelium by transcytosis that does not require the LDL receptor, and they seem to diffuse down and get trapped by proteoglycans secreted by smooth muscle cells, mostly in the deep intima, including by glycan and decorin. Those are negatively charged, and ApoB on atherogenic lipoproteins has a net positive charge. So because of the charge-charge interaction, the atherogenic lipoproteins are trapped primarily in the deep intima and can't leave. And then over time, the smooth muscle cells can take up those lipids. Smooth muscle cells do have scavenger receptors, not as many as macrophages, and the proposed mechanism of lipoprotein uptake by smooth muscle cells is thought to be more by macropinocytosis, which also feeds into the endosome-lysosome pathway. And an earlier study, just getting back to DIT, showed that all humans start to develop this diffuse intimal thickening in atherosclerosis-prone arteries, even beginning in utero. And by the age of 2 years, all humans have this thickening of the intima. And as Dr. Simeone pointed out, the degree of intimal thickening we have is a predictor of future atherosclerosis. So the intima of humans, even before atherosclerosis develops, is basically composed almost entirely of smooth muscle cells and their secreted proteoglycans with very few macrophages. So I think I've basically just said all of the things on this conclusion slide. So smooth muscle cells are by far the most abundant cell type in all stages of human atherosclerosis, likely more than 90%. And macrophages are relatively sparse in early to intermediate athero and then come to be more prevalent in later stages. And smooth muscle cells, and this is a charge-charge interaction for retention of lipoproteins. And so smooth muscle cells form the earliest and possibly the most abundant type of foam cell in plaque. But this had never really been quantitated. And we started working on smooth muscle cells largely to understand how they efflux cholesterol and remove cholesterol. And ABCA1 is the main membrane lipid transporter that helps remove cholesterol from cells, including macrophages. And we wanted to study both medial type and intimal type smooth muscle cells. And human intimal type smooth muscle cells were hard to obtain, they still are. But we had rat medial type and rat intimal type or epithelioid smooth muscle cells. And we found that ABCA1 expression was very low in the intimal type smooth muscle cells and did not go up much with cholesterol loading, whereby it does go up, ABCA1 expression does go up in medial smooth muscle cells when you load them with cholesterol. And then on the right is an image of a human coronary artery. Again, you can see the diffuse intimal thickening, and with Movat's pentachrome, a necrotic core of a lesion. And we did in situ hybridization for ABCA1 and found that it was quite highly expressed in medial smooth muscle cells, but not in intimal smooth muscle cells. So that suggested that if intimal smooth muscle cells are taking up lipids, they would have a hard time releasing it because they have low level of ABCA1 expression, which is a critical mediator of cholesterol removal from cells. So at our site, we are the main heart transplant site for British Columbia, and we have access to many human hearts. All patients who have had their diseased heart removed to receive a transplant have donated their diseased heart to our tissue bank. So Seema Alaverdian in 2014, we tried to quantitate the contribution of smooth muscle cells to the foam cell population. And the green indicates smooth muscle actin, and the red is oil red O. And so we did image analysis with different observers blinded to each other's results. And we concluded that at least 50% of foam cells in human coronary plaque are smooth muscle cell derived. But we were very conservative in that estimate, in that we only counted foam cells that had a high level of actin expression when making this estimate. And we know that many smooth muscle cells in the intima will have reduced actin expression. So this was likely a low estimate. We also found in this study that we confirmed that others had also shown that CD45 is a leukocyte-specific marker. Smooth muscle cells can express macrophage markers like CD68, but to our knowledge, they never express CD45. So CD45 is a very useful way to, when we're doing these kinds of studies, to distinguish the macrophage foam cells, CD45 positive, from the smooth muscle foam cells, CD45 negative. But we found that about 35% of cells that were CD45 negative did express CD68. So that, we feel, represented CD68 expression by smooth muscle cells. So what this means is that we should not use CD68 as an indicator of macrophage presence, because it's also staining smooth muscle cells. So Seema Alaverdian went on to develop digestion techniques where she could gently digest sections of human coronary artery or aorta, and the aortic samples were kindly provided by Dr. Jean-Baptiste Michel from Paris. And so we digested sections of aorta and coronary artery, and then stained the isolated cells with Bodipi, which is a lipid stain, CD45, and ABCA1, and then subjected these to flow cytometry. So Seema first took the cells that had a high lipid content and then analyzed these separately for CD45 expression, and using 11 coronary samples from different patients and 11 aortic samples from different patients, she has concluded now that about 65% of foam cells in the both human coronary and aorta are smooth muscle cell derived, and about 35% macrophage derived, and confirmed also that ABCA1 expression is very low in the smooth muscle cell derived foam cells. There aren't too many other cells that can contribute to the CD45 negative population. We've looked at myofibroblasts and pericytes, and we do not feel they are contributing any major fraction of the CD45 negative foam cells. So Ying Wang in our lab then went on to see what was the contribution of smooth muscle cells to the foam cell population in ApoE deficient mice. And we were not able to perform that histologically for various reasons. So she utilized the same kind of tissue digestion technique. And we used the thoracic aorta of ApoE knockout mice, which after 6 weeks have no lipid accumulation. So they are our negative control. And then the aortic arch for the athero foam cell quantitation. Interestingly, you can see that there's smooth muscle actin in the mouse medial smooth muscle cells, but really almost none in the mouse intimal smooth muscle cells. So mice in particular lose smooth muscle cell markers in the intima, even much more than human intimal smooth muscle cells. So the thoracic aorta cells did not have a lipid high population, so we used that as our gate to count the foam cells coming from the aortic arch, and then we analyzed those for markers of macrophages or not. And we also did lipid quantitation, and the lipid low cells had a low concentration of cholesterol compared to the lipid high cells. And what Ying determined was that about 70% of the foam cells were non-leukocyte in origin, and again, about 30% were leukocyte in origin or macrophage. And this was in non-lineage tracing mice. She also found that ABCA1 expression was also low in the CD45 negative foam cells. And interestingly, she found almost exactly the same results in male and female mice, which is rare and lucky for us. So the reviewers were wanting to know, well, how you have to prove that those are smooth muscle cells. So we used lineage tracing mice, either rainbow smooth muscle cell mice, in which all cells express GFP, but then if you treat them with tamoxifen, the smooth muscle cells lose GFP expression and express one of 3 different colored markers. And that's not shown here, but for simplicity, but the majority of the foam cells were GFP negative and CD45 negative. And again, it correlated very closely to the 70%, 30% ratio, and she used single lineage tracing mice where the smooth muscle cells all express a tomato colored protein, and again concluded that about 70% of the foam cells were coming from smooth muscle cells and about 30% from macrophages. And this has now been confirmed by another group, Mireille-Ouimet's group in Ottawa, who have used PCSK9 gain-of-function mouse model of atherosclerosis and obtained a similar percentage of contribution of smooth muscle foam cells to the total foam cell population. Sorry. So one criticism of this method is that when we digest the tissue, we might be losing one population of foam cell over another. So to test that, we created a collagen hydrogel model where we have layers of collagen and smooth muscle cells and macrophages in co-culture, and then incubate that with aggregated LDL, and then count the foam cells histologically, which is possible in that kind of a model because there's not as big a density of cells, or subject the same hydrogen co-culture gel to digestion the same way we do the tissue, the animal tissues. And we found basically the same estimate of contribution of foam cells to the total population. In this model, it was less because there were fewer smooth muscle cells than there are in human lesions, but the histology compared with our flow method. So we think that this is a reasonably valid way of determining the contribution. So this allows us to compare mouse and human athero. So unless you make mice very severely hyperlipidemic, they will not develop atherosclerosis. They do not have intimal smooth muscle cells. So when you create the severe hypercholesterolemia that's very inflammatory to the endothelium, monocytes bind, migrate in, transition to macrophages, take up lipids, and then cytokines released by the inflammatory cells here cause migration of smooth muscle cells into the intima. And even within 6 weeks of high-fat feeding, when we count the foam cells, 70% of them are coming from smooth muscle cells rather than macrophages. And that has not been recognized in mice previously, probably because, as I mentioned, the mouse smooth muscle cells in the intima have much greater loss of their smooth muscle markers, and they express macrophage markers. So they've been confused for macrophages. Whereas in humans, we all have this diffuse intimal thickening as a pre-atherosclerotic stage in atheroprone arteries, and over our lifetimes, dependent on the concentration of LDL in your bloodstream, much higher in FH, there's a proportional diffusion of lipoproteins into the artery wall. They get trapped by the proteoglycans made by the smooth muscle cells, and you start to develop plaque and foam cell development. So another study had previously this was in 2018, concluded, Kim et al., that using ApoE knockout mice, that macrophages were the main source of plaque foam cells. But what they used to define foam cells was a high level of granularity, which is indicated by side scatter on flow cytometry. So they counted cells with a high level of granularity that were lipid high, but they did not count cells that were lipid high but did not have high granularity. So that didn't correlate with our findings. So what we determined, also published in Ying's paper, was that the smooth muscle cell derived foam cells have low side scatter, shown in blue. And this was using the tomato smooth muscle cell expressing mice. So we found that most of the Bodipi high foam cells had low side scatter, and compared to the side scatter high smooth muscle cells. Foam cells. So that's indicating that for some reason the smooth muscle cells have less granularity than macrophage foam cells. So why do they have low granularity? So lipids are taken up by both macrophages and smooth muscle cells by scavenger receptors, maybe more so macropinocytosis in smooth muscle cells. And all those lipids are transferred to the lysosome, where lysosomal acid lipase cleaves. Most cholesterol and lipoproteins is in the ester form, CE. And that has to be cleaved to free cholesterol for the cholesterol to be able to leave the lysosome. And in macrophages, that proceeds normally, and the increased free cholesterol diffusing to the ER is toxic to the ER membrane, so the ER quickly reesterifies that by the enzyme ACAT and deposits those reformed cholesterol esters in the cytoplasm. And it's those cytoplasmic lipid droplets that are the source of the granularity on flow cytometry, and that's what leads to increased side scatter. So what was the reason the smooth muscle cells have low, the smooth muscle foam cells have low cytoplasmic lipid droplets, basically was the question. So we incubated both human monocyte-derived macrophages or human cultured smooth muscle cells with aggregated LDL, which is thought to be the main form of LDL that leads to foam cell formation in the intima. So we incubated the cells with aggregated LDL for 24 hours. We left the cells without lipoproteins for 24 hours. And then we stained for Bodipi for neutral lipid stain, or LAMP1 for lysosome stain. And what we found was that in the macrophages, by that time, there isn't overlap between the lipids and the lysosome compartment. These lipid droplets are in the cytoplasm. Whereas in the smooth muscle cells, even after allowing them time to catabolize those lipoproteins, there was almost complete overlap between the lipid stain and the lysosome marker, indicating that the lipoprotein cholesterol esters in smooth muscle cells are trapped in the lysosome. And we did this by two different lysosome. We used lysotracker in addition to LAMP1 and found essentially the same results. So we did a bunch of studies of lysosomal function, and we did not find that human smooth muscle cells had low lysosomal function in terms of acidification or proteolysis. But what we did find is that smooth muscle cells have quite a low expression of the only enzyme that cleaves cholesterol esters in the lysosome, lysosomal acid lipase, which is expressed from LIPA, the LIPA gene. And we found that the LIPA mRNA is high in macrophages compared to smooth muscle cells. LIPA expression doesn't go up when you lipid load macrophages, as showing here. Macrophages are, once they differentiate from monocytes, during that differentiation, they develop a high level of LIPA expression in order to be able to receive the substrates that they take up and digest. And we found low LAL protein, low LAL activity, both in the cells and the medium of smooth muscle cells. And then we looked at LAL expression in the human coronary arteries, and I'm not sure how well you can see this, but in the macrophages, CD45 positive, there was a high level of LAL. That's in a section coming right near the endothelium, whereas in the deeper intima, where CD45 is essentially negative, we see less LAL expression, some but less compared to the macrophages. And interestingly, the medial smooth muscle cells seem to have more LAL than the intimal smooth muscle cells. We also found in mice that we didn't have a good mice LAL antibody, so we did in situ hybridization of LIPA in aorta of ApoE knockout mice. And the cells with high LIPA expression were all CD45 positive, as shown here, and cells in the intima that had low LIPA expression did not express CD45. So we think this is a common feature of both humans and mouse, and also the ABCA1 expression is common. So Josh Dublin then treated the human smooth muscle cells with exogenous recombinant lysosomal acid lipase and found that that could cause increased efflux to ApoA1, which is ABCA1 dependent. And so efflux into the medium and reduction of the cholesterol ester pool. And interestingly, the extra LAL had no effect on the macrophages. So macrophages are already making tons of LAL, and if you add more in the medium, it doesn't improve their cholesterol ester hydrolysis. But interestingly, when we increased the cholesterol removal from the smooth muscle cells, we did not see an increase in ABCA1 expression. And nor did we get increased ABCA1 if we added 27-hydroxycholesterol, which is an oxysterol that can directly activate LXR to upregulate ABCA1 expression, and that did not increase ABCA1 in the mouse smooth muscle cells. So we did further gene analysis, and we found that indeed CYP27A1, the protein that makes 27-hydroxycholesterol in the mitochondria, is lower in smooth muscle cells compared to macrophages, and so is expression of LXR alpha. However, if we add LAL to the cells exogenously, it can be taken up, delivered to the lysosomes, cleave the cholesterol esters. That free cholesterol can be released, and that can result in increased cholesterol efflux to the medium with the existing level of ABCA1. But these cells have a different gene expression pattern because they don't release cholesterol from the lysosome, they don't downregulate the LDL receptor, they don't downregulate HMG-CoA reductase. So they don't have the same gene expression pattern as macrophage foam cells. So conclusions from this are that we think that low LAL expression in smooth muscle cells is the natural state of smooth muscle cells. It's not because of a gene polymorphism. LAL has been shown to be GWAS studies, polymorphisms have been shown to correlate with risk for atherosclerosis, but we think this is present in everybody. It's present in mice and humans, that we just all express low LAL in our smooth muscle cells. And that results in the smooth muscle cells that can take up excess lipoproteins retaining the cholesterol esters in the lysosomes rather than in the cytoplasm. So they're a different kind of foam cell. And we think that would make them more resistant to cholesterol-lowering therapies. So even with very intensive statin therapy, ezetimibe, PCSK9 inhibitors, we lower LDL dramatically, but that only reduces recurrent cardiovascular events by 30 to 40%. So this residual cholesterol pool that's in the smooth muscle foam cells of the plaque, we think is resistant to this kind of therapy, and that it may require some way of increasing exogenous LAL to mobilize those cholesterol stores from the smooth muscle foam cells. And we think that our in vitro data suggests that that would happen, and those cells would be able to release cholesterol despite their lower ABCA1 expression. And that could also be by non-ABCA1 dependent cholesterol efflux mechanisms such as passive diffusion, SRB1. Another thing I wanted to address is there such a thing as a smooth muscle cell derived macrophage? And I think we hear this a lot, and I think the answer is no. That even dedifferentiated smooth muscle foam cells, as shown here, they don't have a macrophage, an overall macrophage gene expression profile. They still have a smooth muscle cell gene expression profile. And similar data to this was published by Ed Fisher's group in 2015, that even though smooth muscle cells can express CD68 and some other macrophage markers, they actually have low ability to, they only have about 25% of the ability to phagocytose substrates or efferocytose cells. So I don't think there really is such a thing as a smooth muscle cell derived macrophage. There are smooth muscle cells with some macrophage features, but not truly functional macrophages. So in conclusion, we think that smooth muscle cells are by far the most abundant cell type in human atherosclerosis. Smooth muscle cells form the earliest and most abundant foam cells in human athero and the most abundant foam cell type in mice. And the smooth muscle cells are quite different from macrophages. They have impaired expression of ABCA1, CYP27A1, and LXR alpha relative to macrophages. And they have naturally low LAL expression compared to macrophages, meaning endocytosed cholesterol esters remain in the lysosome. And exogenous LAL can mobilize excess smooth muscle foam cell cholesterol despite their lower ABCA1 expression, and we need to identify the full biologic itinerary of these cells. So I want to thank many students and trainees, postdocs, research associates, and collaborators, including some French postdocs who were in the lab, Kamel Bouquet and Valentin Blanchard. And also thank Jean-Baptiste Michel and Véronique Olivier for contributing aortic samples to this. And with that, I'm happy to take questions and comments.
Thank you very much, Gordon. Is there some questions?
Is that working? Hey, Gordon. Apparently, so cholesterol trafficking is completely different in vascular smooth muscle cells compared to macrophages. Because if there is low, as you show, expression of the LIPA, there is also low few free cholesterol egressors from the lysosome. There is low free cholesterol that's reached the ER. So that might dramatically turn on the cholesterol biosynthesis. So can you see a difference also in cholesterol biosynthesis in macrophages versus...
we haven't looked at that. We have looked at HMG-CoA reductase expression, and it's not downregulated the way it is in macrophage foam cells, and neither is LDL receptor downregulated the way it is in macrophage foam cells. But so I guess your question is, could the increased cholesterol be from increased endogenous synthesis? And I think if it were increased a lot, you would probably see some cytosolic cholesterol ester pool. I don't think it would all go back and reaccumulate in the lysosome, but yeah.
Well, it could also that the excess of LIPA that is secreted by macrophages can enter the vascular smooth muscle cells.
Yes, that's a strong possibility. But as I showed you, most of our sections of human lesions, even advanced lesions, most of the macrophages are just under the endothelium. they're not moving down into the deep intima. So there could be some diffusion of macrophage-derived LAL. I agree. That could be taken up by the smooth muscle foam cells, but that's apparently not happening enough to mobilize the cholesterol from the smooth muscle foam cells. Yeah.
Hi, thank you for the presentation. It was great. I was wondering if you have an idea or hypothesis of what is the part of the endothelial to mesenchymal transition in first the intimal hyperplasia and then to this alpha SMA positive foam cells, because this endothelial cell is going to start to express alpha SMA or other mesenchymal marker.
So is your question, I'm not exactly sure what your question is.
Do you think there is a part of the alpha SMA foam cells that are coming from endothelial to mesenchymal transition?
I don't know if we have good information about that. So you're wondering if endothelial to smooth muscle cell transition is contributing to the smooth muscle population?
Because I guess he has been implicated more, I guess, in mice model in the formation of this intimal hyperplasia.
Mm-hmm.
But I'm not sure if it has been described in the formation of these foam cells. And I don't know if the mesenchymal cells are able to derive into foam cells as well.
Okay, well, I think, yes, in mice, they do secrete, endothelial cells do secrete cytokines that cause smooth muscle cell migration from the medial layer into the intima. And then in the intima, those smooth muscle cells proliferate and start to take up the lipids. In human athero, I think as I showed, the intima is full of smooth muscle cells even before there's any endothelial damage, right? So I don't think it's endothelial. The mechanism of DIT formation is something someone needs to study and win the Nobel Prize for, because if you reduce intimal thickening, you might reduce atherosclerosis. But I don't think the DIT formation is. It could be partly endothelium dependent stress, related flow mechanisms, but yeah.
Okay, one last question. After we will have to move.
So thank you, Gordon, for your great presentation. You're treating a lot of patients with LPL deficiency in chylomicronemia.
Sorry?
Patients with chylomicronemia, LPL deficiency.
That's LPL deficiency, yeah.
LPL, yes, absolutely. Those patients, it was unexpected that some of them exhibit atherosclerosis.
Mm-hmm.
They don't have any cholesterol, but they have very low ABCA1 gene expression, very low ABCA1. Do you think, it's more your thoughts that I'm looking for, do you think that these cases are more kind of a pure smooth muscle cell derived foam cell?
That's an interesting comment. I mean, we don't think that foam cells form because of triglyceride accumulation. That's usually because of cholesterol accumulation, because cells can break down triglycerides, whereas they can't break down cholesterol. Cholesterol has to be removed from the cell, taken back to the liver, and excreted in bile. So why FCS patients develop athero is a good question, and we don't have enough patients with that to isolate their arterial smooth muscle cells yet, but we'll talk more about that.
Okay, thank you very much.
Okay.